Okay, I got a few of you here today. There's a couple things we're going to wander through. When we left off last time, I was working through the details of the muscular system. We're going to, I think, wrap that up today. Remember, I posted that section on visible body. It should be working through that module 13 and module 14 on visible body. And I posted in practice quizzes and a graded quiz for the muscular system. Again, the practice quizzes you can do as many times as you like. The graded quiz is just one opportunity. So make sure you've gone through the muscular system on that visible body before you attempt that one. So with that being said, I think I set the due date for the graded quiz. It's Friday, I think. I think that's what I remember anyway. So you just get that completed before the end of the day, Friday. All right. So with that being said, I want to wander through, kind of pick up where we left off last time, and make sure that we have what we need for the muscular system. We're kind of entering the home stretch here. So wrap up the muscular system this week. Next week, we've got the nervous system. We'll try to get as far as we can in the nervous system, and then it's final exam week. So again, not a whole lot left to do in this summer session. Again, summer kind of goes relatively quickly. It's all condensed into a short time frame. So that being said, let me get into the lecture and where I left off last time. At the end of the lecture, I'll hang out in case there are questions and things like that regarding the material. So this is going to overlap a little bit. If you've already been working through visible body, this will overlap with some of the stuff in visible body, as well as the things I want you to be familiar with. So just any additional stuff. So again, it's all fair game when it comes to then the final exam and whatnot. All right, when we left off last time, if you remember, what I started with is sort of this picture of a muscle. And so when I introduced that picture of a muscle, you should recognize that what we have here is basically a circle within a circle within a circle. Give me just a sec. And so when we talk about a muscle group, remember when we left off last time, what I was trying to get you to understand or see was this entire thing here represents your muscle. All right. So for example, like this entire thing we could say is the biceps brachii or whatnot. So with that being said, though, that muscle is a collection of circles. Those circles that make up your muscle are called muscle fascicles. So each one of these that you have here is a muscle bundle or muscle fascicle. That's what's removed here. If we look within a muscle bundle, all of the circles that you see in here, now it would be this structure that's coming out of here. This is your muscle fiber. So this is one muscle fiber. Remember, muscle fiber is equivalent of one muscle cell. And then if we zoom in on that muscle cell, it is made up of myofibrils. And again, with each of these, remember the number differs depending on the size of the muscle. In other words, we might have one muscle that has 5 total muscle bundles in it, or it could have 20 muscle bundles in it. That muscle bundle could have 15 muscle fibers. So the number within each one is sort of going to vary. So circles within circles. When we had gotten down to that section, then that allowed us to introduce a muscle cell. So now what I have here is, again, this is one muscle fiber. No different than that picture up above, a single muscle fiber. And remember, a muscle fiber is a collection of myofibrils. You are seeing 1, 2, 3, 4, 5 myofibrils within that muscle fiber. So just going back right quick, if you're looking at this, what I was just mentioning, look at the number of myofibrils making up that one muscle cell. The number can vary. I asked you then to wander, essentially working through those parts of a muscle cell so that you were familiar with what it meant to have a T-tubule or transverse tubule, the sarcolemma, a sarcoplasmic reticulum, and so forth. And I think visible body does a nice job of that. where we're going to pick up today a little bit is with this idea of, if you look at a myofibril, along that myofibril, your myofibril is a series of recurring sarcomeres. So I have a sarcomere here. This would be the next sarcomere right here. And it would obviously go that way as well. So along a myofibril, you have a recurring pattern of what's called sarcomeres. It is the sarcomeres that are the contractile unit of the muscle. Well, within this myofibril, making up the sarcomeres, you have more circles. Those are called myofilaments. And there are two types of myofilaments. There are thick myofilaments and there are thin myofilaments. Your thick myofilament is called myosin. That would be some of the circles you see in this myofibril. There are thin myofilaments. Those are called actin. And so making up this myofibril, you have thick and thin filaments called actin and myosin. Now, if we zoom in to see what those filaments look like, then it brings me to my next structure here. So if I was to zoom in, this is then your thin myofilament, and it is called actin. And this is your thick myofilament, and it is called myosin. All right, so making up a myofibril, you have a series of myofilaments, and they are called actin and myosin. And you'll notice here that they interact during a contraction. They grab hold of one another, and that is how your muscle is going to shorten. So there are three main things I want to move through today. All right, number one, we're going to talk about a sarcomere and the idea of the sliding filament theory of contraction. All right. In other words, those filaments that I was just talking about, they slide, they interact, they do stuff when a muscle's contracting. So this explains how that process works. We're also then going to introduce what's called a neuromuscular junction. And we're going to do that so then we can talk about the third thing, which is a muscle contraction. That's the deal for today. That should wrap us up with muscle contraction stuff. Okay, so I have another picture here. And if you look at this picture, then what I've zoomed in on again, look back here, you can tell because there's a sarcolemma there, it is folded back. You can see all the myofibrils and the things laying on top of there. This next picture is a similar structure. Look, there's the sarcolemma, it's folded out of the way. So this is one muscle fiber, again, one muscle cell, all right? This is the sarcolemma, and that sarcolemma, this muscle fiber, remember, is made up of myofibrils. So all those myofibrils within there. Now, what they've removed so you can see it in this picture is, unlike this one up here, they have taken all the structures that would be overlapping or getting in the way out of there so we can just zoom in on a myofibril. In other words, you don't see the sarcoplasmic reticulum and T-tubules and things like that. It would be there normally, but they have removed those structures in this picture so that we can look at the myofibril. Now, if we take that myofibril and zoom in on it, that is the picture that you see here. So now I'm looking at one myofibril, And remember, a myofibril is made up of your thin actin filaments and thick myosin filaments, and they occur in a repeating pattern. In other words, from here to here, right, same thing, you have a sarcomere. So if we were going back looking up in here, you should see that you have a series of repeating or light and dark bands. Those light and dark bands are depicting the sarcomere. In other words, the light and dark bands, this is why we say a skeletal muscle is striated. All right? It is the light and dark areas of this sarcomere. Okay? If we then look at a myofibril is a series of sarcomeres. And something we should mention here is each myofibril has approximately 10,000 sarcomeres. So if I was to look at this one myofibril and count up all the sarcomeres, there are about 10,000. So back to this thing. In a single muscle cell, we have lots of myofibrils. I can't count the number right here, there's too many, but in each myofibril, I have about 10,000 sarcomeres. So let me announce a question I've asked before. All right, I've been told it's tricky. I don't think it is, I think it's specific. Let's go back to here and look at this picture. How many myofibrils are in this muscle cell? I hope you count 5. Now, how many sarcomeres are in each myofibril? Well, this one here would have 10,000. This one here would have 10,000. So if I asked you in this picture about how many sarcomeres are in this muscle fiber, you'd have to say 1, 2, 3, 4, 5 times your 10,000. So it would be about 50,000 in this picture. That is because each myofibril has about 10,000. If we went back up to this picture, how many would be here? Well, there's lots of myofibrils, more than I can count, but a whole lot more than 50,000. Again, but each myofibril, each one has about 10,000. All right, within a sarcomere, I think Visible Body does a nice job of this. Within a sarcomere, you might have done this in lab already. You should learn the parts that make up a sarcomere. You should be familiar with those. All right. Just as a highlight real quick, you should have, give us a little more space here. That making up one sarcomere, you have the area of the Z discs. So a sarcomere is the area between two Z discs. In addition to that, we have the I band. The I band is the area of thin filament only. There is also the A band. Your A band is the area of thick filament. Now let me make something clear there. If you look in this picture, notice your I band technically overlaps two sarcomeres because a sarcomere goes from the Z disc here to the Z disc here, which means I have the next sarcomere starting and going that direction, the next sarcomere starting and going this direction. So when we look at an I band, then the I band or thin filament is coming off the Z disc in this direction and in that direction. That's what you're seeing here in this picture we've zoomed in. There's a Z disc, Attached to the Z disc, you have your thin actin filament. You have your thin actin filament. It is going that direction. Notice something very specific and why we say only. This thin filament, notice, runs all the way to here. So there's a portion of your thin filament that is overlapping the myosin right here. The entire area of thin filament is not your I band. So if again we zoom in on this picture, even though the thin filament continues, the I band is defined as the area of thin filament only. It does not include any of the thin filament that is overlapping with this thick filament. Your I band, your A band is described as your area of thick filament. So it goes from this point all the way to this point, the entire area of thick filament, even that which is overlapping with this thin filament here. Right? In addition to those, you have an H zone. Your H zone is the area of thick filament only. In other words, if we look back at this picture, the H zone that you're seeing right here is the area from the end of the thin filament here to the start of the thin filament here, or by definition, the area of thick filament only. It does not include any of the thick filament over here that's overlapping with the thin. That's the two ways to explain that. Again, as you're going through this, use the picture to help you visualize what these areas are. And then you have the M line. Your M line basically means the middle. All right, it is straight down the middle of your sarcomere, and in a sense, just divide your sarcomere in half. Technically speaking, this M line is where your thick filaments attach. In other words, if we go to this picture somewhere, whoops, the picture right here, if we were trying to depict what that is, my M line would be right here. So if we take this myosin, myosin's been described as like a double-headed, a rod with a double-headed protein. In other words, you can think of it sort of like a two-headed golf club. The handle of it is over here and the heads are up here. And if we think of it that way, the handle of your golf clubs are all sitting here attached to that M line. So if we think of this thick filament as being like a bunch of golf clubs, then the handles of those golf clubs, and you can see that very nicely here. Notice all the heads, you can see the tiny little heads right in here in this picture. Well, all the handles of that golf club or the ends of that rod are sitting here attached to the M line. Right? Those are the details of the sarcomere. And knowing those details allows us to explain something called the sliding filament theory of contraction. In other words, here's what occurs, and it's important again. Okay? When we talk about what happens when a muscle contracts, and when we say a sarcomere shortens, that is absolutely true. A sarcomere shortens during contraction. However, the filaments that make up that sarcomere, the actin and myosin, do not shorten. Students miss that all the time on the exam, and it's very specific and very straightforward. A sarcomere shortens. The filaments do not. You know what the filaments do? They slide. Hence the sliding filament theory of contraction. Filaments slide, and that's how a sarcomere shortens. Now, let me use another example to try to explain and show you what that means. All right, a sarcomere is the area between two Z discs. And that area between the Z discs is made up of those different bands and whatnot. And that's just describing the areas of overlap and non-overlap for those filaments. Okay, so imagine, if you will, that my hands and my wrists represent a sarcomere. And I'm going to show you exactly how this works. Pretend that my wrist is one Z disc and my other wrist is another. So you're looking at a sarcomere right here in the area from my wrist to my wrist. All right? And my fingers represent the filaments. So when a muscle contracts, and I would have a repeating period of this, and then we're looking at a myofibril, when a muscle contracts, that sarcomere is just shortened. Look, the area from my Z disc, a sarcomere, has just shortened. That's not difficult. Now, the question I ask here, have my fingers gotten any shorter? No, my fingers are the same length. They're not suddenly chopped off. All that happens is they slide, so the amount of overlap changes. The filaments don't shorten. The sarcomere shortens. So what the A band and I band and all of that stuff does, it allows you to explain how those areas change when the filaments slide. Which brings me back to the picture that you see here. Notice, this is when your muscle is relaxed and not shortening. And so what I have here is a Z line or Z disc. This would be the other Z disc or Z line, which means that is your area of a sarcomere. Now, what they're not showing you is straight down the middle here. We have our M line that would divide this thing in half. We've already seen now that I've got an I band. So this whole area right in here is your I band, the area of thin filament only, doesn't include any of that thin filament that overlaps. And then we have the area from here to here, and that is your A band. And then the area from the end of the thin filament here to the start of this next thin filament, or the area of thick filament only, that's your H zone. Now, if we look at what happens during the sliding filament theory of contraction, then the picture down here now shows when the muscle contracts and a sarcomere shortens. Notice the filaments aren't any shorter. They simply are overlapping more. So what changes in the sliding filament theory of contraction? Here we go. There are several things you should be able to say. In the sliding filament theory of contraction, do the Z discs get closer together? Well, here's your Z line or Z disc and Z disc. Yes. So your Z discs move closer together. Does your I band get shorter? Well, look, that was the area and this was the area of your I band. Is it much less? Yeah, now that's the area of your I band, I band. So the I band gets smaller. Does your A band change? look, here's our A band, the point from here to here. And if we go straight down to this next picture, no, that does not change. The A band stays the same. Well, what then happens to your H zone? Your H zone, that area of thick filament only, it was this wide when your muscles relaxed. Now it's this wide. So it has gone to this point, which means your H zone is smaller. It may even disappear if it moves close enough together. Does anything happen to your M line? No, the middle's still the middle. Think about that golf club analogy and the handles of your golf club being attached to that M line. When you swing your golf club, the handle stays in your hand, or it should. So it didn't go anywhere. The end of the golf club may move, which means, again, the M line's not changing. Back to this picture I was trying to show you back here, the head of our golf club sitting there, all the handles, or in the picture right here, the head can move just like the end of your golf club. The handle stays over here, so your M line's not changing. Okay. Sliding filament theory of contraction. It wanders through the details and takes a sarcomere within a myofibril and explains how those filaments change. Now, when that occurs, keep in mind all of the sarcomeres within that myofibril are going through that process. All right? So those heads that I was showing you, those golf clubs, imagine instead it's like a bunch of people in a boat rowing a boat. All right? Well, everybody's doing something, sticking the oars in the water and rowing along. Same thing happens with those filaments in that myofibril as a sarcomere shortens. Does that make sense? It's a lot, but you should be able to work through both visible body and that picture I provided and try to understand what that's meaning when those filaments are working. If you have questions about it, reach out and let me know, and I'll hang out at the end to try to address those. I next am moving on to the parts of a neuromuscular junction so I can walk through the process of a muscle contraction. This is done very nicely on visible body, so I'm just going to hit some highlights of that to make sure that we're all on the same page as we work through the weekend trying to get muscles out of the way. Okay. A neuromuscular junction, by definition, is where a neuron meets a muscle fiber, or the area where a neuron meets a muscle fiber, where a neuron meets your muscle cell. Now, there's some other things that I want to make clear as well. There is only one neuromuscular junction for each muscle fiber. In other words, you'll only have the end of a neuron touching a muscle fiber one time. Let me try to explain what I mean there. If we look, hopefully you recognize that you have a muscle. Like your big giant biceps. And that muscle is made up of all those muscle bundles, and those muscle bundles are all made up of muscle fibers. So right here, each of these represents a muscle fiber, a muscle fiber, a muscle fiber, a muscle fiber. All right? So you have lots of muscle cells within, say, for example, your biceps. All right? Well, what I'm saying here is that a neuron, oops, wrong color. A neuron, it may branch. That's great. Think about it like a tree with all the branches. This muscle fiber may talk to this muscle fiber. This neuron will talk to this muscle fiber. It will not come over here and talk at another point. All right, this may branch and have another neuromuscular junction over here. And another one that's over here. But you will not have that occur. There is only one neuromuscular junction per muscle fiber. All right? A neuron can talk to more than one muscle fiber. So this neuron may branch and talk to more than one muscle fiber. That's what a motor unit is. A motor unit is defined as the neuron and all the muscle fibers it talks to. But you will, for each muscle fiber, only have one neuromuscular junction. This idea helps to explain how when you contract your biceps, you can get contracted to different degrees. And I'm going to come back to this concept. Let me show you an example. Dr. Grillo is going to work out for a second. And I'm going to use my biceps to curl this here pen. And you see my biceps curl. All right. Now I'm going to pick up something that's heavier. And I'm going to use my same biceps and I'm curling a big giant bag of oranges. Okay. Do they weigh the same amount? No. Am I still using my biceps? Yes. What's the difference in those two things? Well, it's how heavy it is. Well, what it relates to is I use the number of motor units required to lift what I'm trying to lift. All right? Which means, if we go back to this concept, within your biceps, you have several motor units. This may be motor unit number 1. I may have a second motor unit that comes over here and it talks to this muscle fiber and that muscle fiber. This would be motor unit number 2, the neuron and however many muscle fibers it recruits. So if I need to lift up something really light, I may only have to recruit one motor unit. If I lift something more, I may use both of these motor units to contract. All right? And obviously, within a muscle, you have a limited number of motor units, which essentially means you have a limit in how much you can lift. Other words, I could lift this house if I just recruited more and more motor units. I can't do that. I can use the number of motor units I have, all right? And that then describes what I may potentially be able to lift. That is the difference in a motor unit and a neuromuscular junction, something that confuses students often. A motor unit is a neuron and all of the muscle fibers it innervates. Notice in my motor unit 1 neuron is talking or has 3 neuromuscular junctions. Motor unit 1, that neuron is talking to these 3 muscle fibers. Motor unit number 2 is talking to these 2 muscle fibers. But each muscle fiber only has 1 neuromuscular junction. It only has 1 point where that neuron meets the muscle fiber. Or you may see it as where 1 axon meets the muscle fiber. all right? Because the point where a neuromuscular junction is the end of your neuron. So if we zoom in and look at that point right here, then we're looking at a neuromuscular junction. So what we have here is, imagine I have my neuron, and I come to the end of that neuron. The end of that neuron is going to meet my muscle fiber. So technically, this whole thing right here represents the neuromuscular junction. This would be the axon of my neuron, and this would be the axon terminal, the end point of my axon. And this would be my muscle fiber. And so this line right here would represent my sarcolemma, or the cell membrane of my muscle fiber. And so this is essentially your neuromuscular junction. All right. Now there's some parts. I want to make sure we know all those parts. Okay. This area where you have the axon terminal meeting your muscle fiber, there's a gap. This is called the synaptic cleft. It is the area between the axon terminal and part of your sarcolemma that's indented. This portion on your sarcolemma that's indented is called the motor end plate. Notice something very specific here. Your axon or neuron does not directly touch your muscle fiber. There's a space between them. That is called the synaptic cleft. In other words, this neuron doesn't ever touch the muscle fiber. All right? That means when we have when we have a muscle contraction, what we're going to do is we're going to have an electrical event on your axon. A voltage travels down it. That electrical event has to become a chemical event in this gap in order to connect the information from this neuron to your muscle fiber. So there's an electrical event that will generate a chemical event so that we can create another electrical event on your muscle. Those steps are going to lead to a contraction. All right, so we need a couple of more things. Go away. Oh, well. All right, there are some other structures that we need to know. And in this axon terminal, there is a structure here, and it is called a voltage regulated calcium channel. There's a voltage regulated calcium channel in your axon terminal, or I should say in the membrane of your axon terminal. In addition to that, in your axon terminal, there are synaptic vesicles. In those synaptic vesicles, there's something called ACH. And ACH stands for acetylcholine, right? Over here on your motor end plate, see if I can get that out of the way here. Over here on your motor end plate, we have a structure and it's kind of tied together. Some books do them as separate, but on here, we have an ACH receptor, and that ACH receptor is tied to something called a chemically regulated sodium channel. Right? Now, these are all the parts that allow us to kind of walk through the initial steps of a muscle contraction. Again, Visible Body does a nice job of walking through this process. I'm going to hit the highlights of some of that right quickly here by drawing that neuromuscular junction again. If we talk about a muscle contraction, I am very quickly going to just talk about the initial steps of what occurs and what Visible Body should say. And here we go. An action potential travels down the axon of your neuron and reaches the axon terminal. Your action potential is the electrical event, the voltage that I was talking about a minute ago. So that electrical event is an action potential that travels down your neuron. When it reaches the axon terminal, the action potential triggers your voltage regulated calcium channel. Now, this goes back to something we did early on. I had you learn the ions and their concentration inside and outside the cell. So if we go back to that idea, then I hope you recognize that all of the area out here is the ECF, your extracellular environment. So you should know the concentration of the ions in that extracellular environment. It helps explain what occurs here in this next step. When an action potential triggers this voltage regulated calcium channel, you know what it does? The calcium channel opens. And when that calcium channel opens, I hope you remember that calcium is high outside the cell. So when that channel opens, calcium goes flooding in to your axon terminal. And when it goes into there, that calcium binds to the synaptic vesicle, which releases the ACH out into your synaptic cleft. All right? When that ACH is released into the synaptic cleft, I hope this next step should be obvious. That ACH goes and binds to your ACH receptor. The ACH receptor on the motor end plate is a receptor for the ACH. When it gets released, it binds to that ACH receptor. Binding of the ACH to that receptor is what's going to open the chemically, sometimes Invisible Body may call it ligand. Ligand means chemical. But ACH binds to that receptor, and the binding of the ACH, the binding of that chemical, is what opens the chemically regulated sodium channel. So when ACH binds to this receptor, that ACH causes this chemically regulated sodium channel to open. And what happens to sodium? Well, if we remember again, back to our concentration gradients, sodium is high out here in the ECF. So when that chemically regulated sodium channel opens, sodium rushes in to the sarcoplasm of your muscle fiber, and that rush of sodium in, the rush of those positive sodium ions in here, is what causes your next electrical event. It generates another action potential on your muscle fiber, right? It is this action potential that then goes and travels along here. And when the action potential travels along there, that action potential then goes and travels down the transverse tubules. That again is a structure you should have learned back up when you were looking at that muscle way back here. Again, I asked you to do this on visible body. Way back here, the T-tubule, the transverse tubule. The job of that transverse tubule is when an action potential occurs and travels along that membrane, it goes down the T-tubules. In other words, it can spread through the muscle fiber. That's what the point of a T-tubule is. It carries the action potential deep into the muscle fiber. It carries the electrical event or the voltage down into the T-tubule. All right? If we pick up at that T-tubule then, all right? If I look along that T-tubule, we have another channel. In the T-tubule, we have a second voltage regulated calcium channel. So when this action potential travels down the T-tubule, it is going to trigger a voltage regulated calcium channel again, a second one. So you have two areas where you have a voltage regulated calcium channel. You have one in the axon terminal and you have one down at the T-tubule. All right. Triggering of this voltage regulated calcium channel is what then causes your calcium to come out of the sarcoplasmic reticulum. So again, structures I asked you to learn and spend time with. If you haven't done this yet, a lot of it will be confusing here, but back to this picture. All right, this area here leading over to this is your sarcoplasmic reticulum. And that sarcoplasmic reticulum, SR, the job of that is it stores a high concentration of calcium. So sitting inside of your muscle fiber, you have the sarcoplasmic reticulum, and in that sarcoplasmic reticulum, you have calcium. So when an action potential hits this voltage regulated calcium channel, it is what causes the calcium to rush out of your sarcoplasmic reticulum. And again, we're sitting into the sarcoplasm. That is then what initiates the next step of a muscle contraction. And it shows that in visible body as well. Okay. If a lot of that sounds new because you haven't worked through visible body, then you need to start doing that and then come back to these notes so that you understand this process of a muscle contraction, sliding filament theory, and then the steps that are going on as a muscle contraction takes place. I want you to do that for the rest of this week, all right? Because next week we're starting the neuron, okay? I think that's probably enough for the day. That's a good bit of detail. But again, if you use this in conjunction with visible body, then you should be able to work through the parts of your muscle and most of the steps of a muscle contraction. All right, I'll stop the recording there. I will hang out in case people have questions.